family tree that is Linnaeus's hierarchical system that you probably talked about in Core 1, that is the kingdom, phylum, class, order, family, genus, and species, has been modified a little bit by adding the domain at the top. So in other words, they have a larger group in which you have kingdoms underneath that. And I'll talk about what that is in just a second. Just by the way, the classification system in Appendix E is what we're going to be using for this class. So if you're confused or you're having trouble trying to figure out what's what, that's where you look. It's make sure you kind of check it out right now. Prokaryotes are like the two big domains, that is archaea and bacteria. There are some single-celled eukaryotes as well that we call protists, and we're going to go through those. There's actually many of them, many kingdoms within that group. But first we're going to talk about the bacteria, but I wanted to just show you first, this is how the domains kind of break out. In other words, the domain bacteria is going to have these groups, and we'll talk about these. The domain archaea has these, and all of these guys are considered what they call extremophiles. In other words, high methane conditions, halophytes are high salt conditions, thermophiles are like in hot springs, like say Grand Prismatic Springs or something in Yellowstone Park. The eukaryotes, of course, are everything else. So these are all the organisms that have a nucleus, etc., of which we have the protists, like I just mentioned, but we also have fungi, plants, and animals. All right, so we will go through all of the diversity of that, but we're going to focus on the bacteria first. I gave you in your handouts, but this is also in your textbook, this table that talks about the differences between bacteria, archaea, and eukarya. It used to be thought that archaea was part of the bacteria prokaryote group, and they all used to be lumped. But if you look down this list, you can see that there's quite a bit of differences between the two groups. Sometimes the archaea is more like the eukarya than the bacteria are. So anyway, that's something to take into consideration when you're looking at those things. Okay, so first of all, we're going to be talking about the bacteria. Bacteria are prokaryotes, and prokaryotes, recall, just have a cell membrane, the circular strand of DNA, and then they have ribosomes that do their protein synthesis, and they have some cytoplasm. And of course they're small, and another big thing is that they have a cell wall. So all current bacteria will have a cell wall. So we're going to talk about what those are in just a second. So there's three major shapes of bacteria. There are what we call, I thought I had a overhead of that, I guess I don't. They're rods. And you might recall this again from Core 1 because we looked at the bacterial shapes. They are rods, or that is kind of sausage shaped. They are circular, called cocci, or they're kind of a corkscrew shape called spiralis, which are generally like really thread-like and small. They also have a cell wall. The cell wall has some differences though. So some of them have the cell wall, and it has a peptoglycan layer. So if you recall the terminology from these organic molecules, pepto means that it's got proteins, and glycan would mean that it has some kind of polysaccharide. So it has a peptoglycan layer on top of that cell membrane. These are what we would call gram-positive, because they would take up gram stain. Now the ones that are what we call gram-negative, they have also the peptoglycan layer, but they also have a membrane or like a layer that is outside of that has lipopolysaccharides. Lipo means that it has a water repellent layer, in other words, lipids that are part of that. And what that means is it's going to repel the gram stains. So these would be what we would call gram negative. So just backing up here, gram negative and gram positive. Also, the cell wall can have some other features, like some of them have something called a capsule. This is just like a sticky surface. It could be a protein or a polysaccharide, and it allows cells to stick together, or it could like stick to a host cell or something like that. And some have fimbriae or pili, which are just appendages. They are not flagella or cilia, although they might kind of look like that a little bit, but fimbriae are short and pili are long. They do, some of them, more than half of the prokaryotes, that is bacteria, have a flagella. These flagella operate a little bit differently than eukaryotes. In fact, they actually look quite a bit different. And this is also a picture in your textbook. Whoops, upside down and backwards. Okay. So they can have more than one. This picture is showing 3. Usually it's 1 or 2, but sometimes more. And they have, like, literally, it looks like a little rotor here, and it actually operates quite a bit differently by turning around as opposed to swishing back and forth. The eukaryote flagella would allow more directed movement, although these guys, do fairly well. It's just a slightly different operating system. It operates by pumping hydrogen across the membrane and then letting it flow back across. That's the energy source. So it uses ATP to do that, and just the system of that, it just looks a little bit different. Now the reproduction is done via binary fission. You might want to review that. I think it's in chapter 12, not really, I can't really remember exactly, but you might want to review. And the thing about the internal structure, the part that gets reproduced, or that is part of the part of the DNA part, is the nucleoid. That's a single strand of DNA. The actually reproduction of that single strand is similar to the actual replication of a chromosome, slightly different in terms of the kind of enzymes and so forth used. But they also have another smaller genome groups that are called plasmids. And there may be different plasmids in different bacteria, even of the same species. These plasmids are thought to cause either nutrient or help with nutrient use in a specific location, and or give resistance to things that might be attacking it, such as antibiotics or something like that. So that might be something that's important to antibiotic resistance. They can also form something called an endospore. An endospore is formed when conditions are super harsh. And this is basically just kind of a dormant structure that allows it to stay in a dormant state, and it can be up to a century, so a fairly long period of time. They also have something called horizontal gene transfer. This is where the plasmids from one group, it might be within the same species. They might have one type of plasmid because of where they are existing. And they could, when they do the binary fission, these plasmids that might exchange information and give, some of the cells might have more of one type than another, and that might make some slight differences between the groups, even though binary fission is really a lot like mitosis, all right? And they also can do something called transformation. This is, they incorporate some DNA from the environment. So random, pieces of DNA that might have gotten broken down from the digestion of an organism or something like that could be taken up. And they might also do something called transduction. And this is where viruses might exchange or give some of their genome to the actual bacteria cell. So even though they reproduce by binary fission, which is basically like mitosis, it doesn't mean that they are all alike. And in fact, they can change based on their environment, and also things like viruses that might be attacking them.